qPCR, or quantitative polymerase chain reaction, is a method for measuring water contamination that can provide same-day results while keeping overall lab costs nearly identical to former methods after the initial capital investment. Enterococcus bacteria, found in the feces of most warm-blooded animals, indicates pollution of waterways with human pathogens. Enterococcus is expected to become the new federal standard for water quality at public beaches because it is more highly correlated to the human pathogens found in city sewage than fecal coliform. In the past, the predominant method of testing for fecal indicators involved culture-based methods, which could take as long as 48 hours to accurately measure. The qPCR technique can give results in as few as 3 hours, allowing warnings to be posted and infected waterways to be closed for cleanup the same day the samples are collected. Samples are taken using sterilized bottles to minimize cross-contamination of the DNA from other DNA sources. The sample collector carefully wades into the waterway to avoid kicking up sediment and plunges the bottle straight down to avoid including surface scum. Samples are then transported to the lab in coolers at 4 degrees Celsius. PCR, or polymerase chain reaction, replicates many copies of DNA from a single molecule. Quantitative PCR can accurately predict the original amount of Enterococcus DNA from the number of DNA copies made. The lab technician first filters the sample to concentrate the DNA, then extracts the DNA from the filter by beating and centrifugation. In order to maintain experimental control, blank filters without DNA are also extracted, along with known amounts of salmon DNA for reference. This ensures the results have not been contaminated during the process. The EPA is moving towards Enterococci qPCR testing for 2 reasons. 1, because Enterococci is more closely related to human illnesses from recreational waters than E. coli. And 2, because results are obtained in as few as 3 hours as opposed to 2 days, allowing for faster corrective action to be taken, and as a result, safer public waterways.